Today we're going to go ahead and move on past our chemistry chapters and get into cells. And remember, cells, we said, were the building blocks of life. All living things are composed of cells. So today we're just going to introduce a little bit about cells and then cell structure here in chapter 4. And we did mention the cell theory earlier in the semester when we talked about our characteristics of life. Now it is a theory, so we know it's very well proven. It's been very thoroughly tested over time. And our cell theory has three aspects to it. First, all living things are composed of cells. Our second aspect is that the smallest unit capable of life then is a single-celled organism. And then finally, all cells come from pre-existing cells. So again, when we think about living organisms, we are all built of cells, and it certainly is critical to understand what makes up a cell and what are some different types of cells if we're going to understand life. The first thing that we kind of notice if we're going to start looking at cells and studying cells is we can't see them. They're microscopic, so we have to have our special tool, our microscope, to be able to see cells and to be able to study them. And so, not surprisingly, our first cell was discovered and described about the same time we got our first microscopes. And the first light microscopes were invented in the middle of the 1600s, around the 1650s or so, about the time when physicists were getting good at grinding glass lenses. We see our first microscopes. We also see our first good telescopes as well. And Robert Hooke, who was a physician in England, was the first person to describe a cell. He happened to be looking at bark, tree bark, underneath the microscope, and those cells are dead, and so it looked like an empty container or an empty cell. That's where it got its name. And certainly much of what we've learned about cells has occurred using the light microscope, using our basic tool for well over 300 years. And so we'll take a look at our light microscope, and certainly we'll use them in lab and talk a little bit about the properties of the light microscope. More recently, we have electron microscopes. They first appeared last century. Today we have many, many different types of electron microscopes. These electron microscopes give us a whole new view on life and a whole new view on cells, and we certainly learned an awful lot in the last 40 or 50 years using our electron microscopes. So I just want to start off with a little comparison between our two different types of microscopes and what we're going to see, what we can hope to learn by using those different tools. I'm sure you've all used a light microscope somewhere in the past, whether it was high school or another college course perhaps. But here we can see a typical light microscope. The light microscopes today are called compound light microscopes because they have multiple glass lenses that you look through. And if we look on the left there, the little picture shows how we're going to view things under the lenses. Light source at the bottom, it's going to shine up through our microscope slide, and when we view things we're looking through two different lenses. We're looking through the ocular lens, which is where the eyepiece is, and then we have that objective lens, and that's the one you can change to make the magnification larger or smaller. And really our basic tool hasn't changed much in well over 100 years. There are many advantages to the light microscope. One is the idea that in lab we'll be able to pick one up, and within 15 or 20 minutes we'll be able to use the light microscope as a tool. So it's easy to learn, and that certainly is beneficial when we start thinking about labs and learning about cells. But in terms of what we learn about the cells, we can observe cell shape. We can observe different types of cell, and so because our light microscope's been around a long time, for example in humans we know we have over 100 different types of cells, and they've been described in detail for a very long time. And we can observe things while they're alive, and again we've all done probably somewhere along the line the put a drop of pond water under the microscope trick, and you can see how amoeba ooze around or paramecium swim and so on. So these are certainly all nice advantages to using a light microscope. There is, of course, a drawback to the light microscope, and the major drawback or disadvantage to the scope is that we're limited by how big we can magnify things. And light microscopes have a limit about 1,000 times magnification, meaning we can look at things 1,000 times bigger than they really are. And if we try to magnify things larger than that 1,000 times, we can make things look bigger, but we lose resolution. We can't focus it anymore, and so that's a very fixed limit. And that limit again allows us to see cell shape and type and so forth, but it doesn't allow us to see inside of cells very much. And so using the light microscope we can see the nucleus, we can see a couple other pieces of the cell, but most of what's inside of the cell has remained a mystery until our next tool came along, the electron microscope. And here's a picture of a typical electron microscope. Electron microscopes have a lot of computing power associated with them. They use a beam of electrons that they're going to shoot toward the target. Those electrons bounce or scatter off, and then there's a field that measures where the electrons bounced off to, and based on the pattern of electrons, the computer then is going to generate an image that we've seen. And so electron microscopes are extremely expensive. They're really large. They're very hard to use. It takes a lot of training to use them, but they are going to give us the first chance to peer inside of a cell and see what is a cell made of. And that's the major advantage of the electron microscope, is electron microscopes have a much higher magnification, and a much higher resolution. We can blow images up much larger than they are in real life and still have that fine resolution to be able to focus them and see lots of detail in terms of structure. And today, current electron microscopes have a maximum of about 100,000 times magnification compared to real life, which is going to allow us to see inside of cells again for the first time. We can look at cell parts. And in fact, electron microscopes, as our technology and computer power certainly gets better, what we're seeing is this magnification limit is going up with electron microscopes. This is not a fixed limit, but it's getting higher. And certain electron microscopes today, even, people are looking at chemicals and looking at the shapes of proteins or the shapes of DNA. And so theoretically, we'll be able to look and magnify all kinds of chemicals all the way down to an atom eventually to see what shape an atom might be. Now, the major disadvantage as we look at the electron microscope, as I mentioned before, they are pretty expensive and hard to use, but also we cannot see organisms while they're alive. We have to treat the specimen to be able to use the electron microscope. It's going to kill the specimen, and even if it didn't, if we shot a high energy beam of electrons into the specimen, it wouldn't live long anyway. And so the drawback here is we cannot see organisms while they're alive, while they're active. So if we just compare some images that we might see, here's a picture from an electron microscope of a plant cell. And if we look at the plant cell here, the big open area in the middle, that's a vacuole that's usually filled with water that plants need for photosynthesis. We can see three big chloroplasts spread out around the edge, and then the nucleus there is kind of smashed between one of those teardrop-shaped chloroplasts and that big central vacuole, that big kind of empty space in the middle. And so, again, our electron microscope allows us not only to see the shape of this plant cell, but we start to see structures and details and parts inside the plant cell. And if we just do a quick comparison, here's a picture of some cilia under the light microscope. We can see these little single-celled protists are kind of almost spaceship looking, and they've got a little ring of cilia or eyelash looking structures around the edge. These things beat, and that's how they swim along. So using the light microscope, we can see the little eyelashes. They look kind of clear. We can see they're there, but we really can't see much about them. If we look at cilia, though, under the electron microscope, what we see is we can blow them up big enough, we can see individual hairs. We can count the cilia. We can see how they're attached or how they're arranged. And so again, it's given us a whole new view on cells and that microscopic world by using the electron microscope. And certainly much of what we talk about in cell structure in Chapter 4 is based on information from the electron microscope. And as humans, as adults, we're made up of 100 trillion cells. And if we write that out, that's one and an awful lot of zeros. And in fact, this number gets so big, it's kind of meaningless because it's such a large number. But as we talk about cells and microscopes, it kind of begs the question, why do we need microscopes to study cells? Why are cells so small? Why are they all microscopic? And this isn't something just in humans or in animals, but this is across the diversity of life. That whether I'm looking at bacteria, or those archaea living down by thermal vents, or I'm looking at fungi, cells are microscopic. And so anytime we see a pattern like that that's broad across the diversity of life, we know there must be some underlying factor that's keeping cells small, that's preventing cells from growing very big. Okay? And just as an example, we're made up of those 100 trillion microscopic cells. Why don't we survive with 100 million bigger cells? Okay? But we don't do that. The cells, again, don't grow bigger. They seem to be restricted in terms of their size. And as we think about that question, what factors might keep a cell from growing, again, not just in an animal, but in all types of organisms, the main common denominator is an idea called the surface area to volume ratio. Okay? And it turns out this is probably more of a geometry problem than it is a biology problem. That as we look at cell size is going to be limited by this concept of surface area to volume ratio. So I want to take a look at, first of all, what the heck is surface area to volume, and then more importantly, how would that limit the size a cell may grow? So if we take a quick look at a cell, the surface area is simply the part of the cell that comes in contact with the outside world. So we can think of it sort of as that outside edge of the cell. Again, it's going to separate inside from outside. And certainly cell membranes are going to form this surface area that's going to allow contact with the outside world. And the major function of this surface area is going to be exchange of materials. Okay? Getting food in, getting oxygen into that cell, if we're dealing with an animal cell, for example, getting waste products out. And so constantly living things have to exchange materials with the surrounding world to remain alive. Okay? The volume then is basically everything else inside of the cell. Okay? Okay? And this is, we can think of this volume as sort of the needy portion of the cell. This is the part of the cell that needs food to stay alive. It needs to get rid of the waste. And so we've got these two kind of contradicting aspects of the cell. The surface area is providing the exchange of materials. The volume needs that exchange to stay alive. And if we look at that electron micrograph of that plant cell we mentioned before, and this is 2D, so we need to think 3D, but here we can see the surface area is that outside edge, and then the volume, everything in between. And so anything that gets into or out of the cell has to be able to cross that surface area. So let's take a look at the surface area to volume ratio and how it is going to change based on cell size, why this might be important in terms of how cells grow. So let's take a look at a cell that's relatively small. Here we'll take a cell, and I looked at a cube or a square-shaped cell. Think of a box or maybe a die, because straight lines and squares make math a little bit easier. And so in this case, we're going to take a look at a cell that's 1 millimeter wide, 1 millimeter tall, and 1 millimeter deep. Okay? So the surface area to this particular cell is basically going to be that outside wrapping of the box. And so to figure that out, we just need to figure out what's the area of one of the squares. And since a box has 6 sides, we'll take it times 6. So in this case, the area of one square is going to be the height times the width, or 1 millimeter times 1 millimeter. And then we'll take that again times the 6 sides of the box. So our surface area for a box that's 1 millimeter by 1 millimeter by 1 millimeter would be 6 squared millimeters. The volume then we just figure out that's the height times the width times the depth, or in this case 1 times 1 times 1. So this particular cell is going to have a surface area to volume ratio of 6 to 1, meaning for every one volume inside that needs food, it needs nutrients, there are 6 units of surface area to do that exchanging, to keep the volume fed, to remove waste, to exchange gases and water, and so on. Now let's take a look at what happens to that surface area to volume ratio if the cell is larger. Okay? So let's again stick with our square-shaped cell, but this time let's just double our measurements. We'll make the cell 2 millimeters wide, 2 tall, and 2 deep. So our surface area again is going to be one side and then times 6, and so the surface area of one side of the box is going to be 2 times 2. So we end up with a surface area of 24 squared millimeters. Not a big surprise, we've got a bigger cell, we've got more surface area. Likewise, our volume, we'll take our height times our width times our depth, so we're going to get a volume of 8 cubic millimeters. Again, not a surprise, we've got a lot more stuff inside, we've got a lot bigger cell. But notice if we compare our ratio of surface area to volume now, it's 24 to 8, which factors down to 3 to 1, which means for this larger cell, even it's the same shape as that small cell, but just by being larger, this cell has only 3 units of surface area to keep every unit of volume alive inside. Okay? So a cell that's this large would not have enough surface area to keep the volume fed, to get rid of waste and do the exchange. And so what we would see is as the cell gets too big here, it's not going to be able to survive because it doesn't have enough surface area to feed that volume. And so while cells get larger, surface area and volume are going to get larger, but not at the same rate. The volume is going to increase much faster than the surface area. So our take-home message here with our surface area to volume ratio is that as cell size increases, as our cells were getting bigger, that surface area to volume ratio decreases. It's getting smaller. So one of the reasons, in fact the main reason that cells are microscopic, is if a cell gets too big, it won't have enough surface area to feed and to keep that volume alive inside. And so that's the main restriction we see on cells, and this is why this restriction is across the board of diversity of life. Again, whether it's a plant or an animal or a bacteria, because all cells need to exchange materials with the outside world. If they get too big, they simply can't do that fast enough. Now your book looks at the same problem a little bit differently, but comes to the same conclusion. In your book it talks about having one large cell here, like on the left, or if we took that same area and divided it into 27 small little cells. And the small little cells have a lot more surface area than that one large cell. So again, that's the benefit of cells being microscopic. As we look at these microscopic cells, one thing we see that's pretty amazing is there's a tremendous amount of cell diversity in terms of the shapes, the types of cells, how cells go about life. And we can split all cells into 2 main categories. And we mentioned these when we went through our diversity of life a few days ago, that one group of cells are the prokaryotic cells. And remember we said prokaryotic cells are relatively small and relatively simple. They don't have a lot of different cell parts. And our defining feature of our prokaryotic cell is that they don't have a nucleus. Okay? Their DNA is mixed in with the rest of the cell. Not only do they not have a nucleus, they don't have many other cell parts. When we look at them with an electron microscope, we don't see a lot of structure. Okay? And remember, prokaryotic cells make up two of our three domains. They make up the archaea and the bacteria. And that we only find prokaryotic cells living as single-celled organisms. We don't see any multicellular life with prokaryotic cells. We only see single-celled life. And these are certainly by far the most ancient form of life on our planet. Our oldest fossils date about 3.5 billion years old, and they're made up of entirely single-celled prokaryotic organisms. In fact, all of the fossils on our planet that are over about 2 to 2.2 billion years old all contain nothing but single-celled prokaryotic organisms. So we know that the first forms of life on our planet were all these prokaryotic organisms. And if we take a look at some examples of diversity, here's a bacteria cell under a microscope, a light microscope, and we can see it's kind of shadowy there. We see these long spicules or spikes. These are actually little hollow glass tubes, like little glass hypodermic needles. This is a predator. And when other organisms swim by, they get impaled or stuck on those needles, and they will use those needles then as a straw to suck out the cytoplasm and the energy of another cell. Okay. Here we can see some other bacteria. This is probably more of a typical bacteria. The round shape is looking at the end. It's an oblong or sort of pill-shaped cell there. The clear all throughout that cell is actually the DNA. So that's the genetic material kind of mixed in with the rest of the cell. We can see they have a big thick cell wall or barrier on the outside there, that kind of very dark rim around the bacteria cell, but not many other cell parts. We don't see a lot going on here with the bacteria. And many of the bacteria may be photosynthetic, and they may live in groups. And so here we see several different types of bacteria, all green, indicating photosynthetic. Oftentimes they do grow in these long chains, and so when we look at these chains, each little dot is a bacterial cell, and so they may be 40, 50, 100 bacteria cells all growing in these very long photosynthetic chains. Our other major type of cell then are eukaryotic cells. And by comparison, our eukaryotic cells, which certainly is our cell type, are much larger and more complex than the prokaryotic cells. Not only do we have our DNA in its own little compartment in its nucleus, but we've got a lot of other cell parts as well. Our cells are much more complex. Remember, our cell type, eukaryotic cells, are only found in the domain eukarya, which includes our four kingdoms, the protists, the plants, the fungi, and the animals. And while some organisms are single-celled eukaryotes, most eukaryotes are multicellular. And we know that our cell type has been around certainly for a very long time, about 2 billion years. Okay? But that leaves about 1.5 billion years where all life on our planet was only prokaryotic. So by comparison, eukaryotic cells are much younger. And much like our prokaryotic cells, an incredible amount of diversity when we look at eukaryotic cells. This is a typical paramecium we might pluck out of pond water, and we can see right away, look at all the different cell parts. The large spidery looking structures, there's one at each end of the paramecium. Those are called contractile vacuoles. They act as water pumps, constantly pumping water out of the cell, otherwise it would swell up and burst eventually. The nucleus there is right in the center, and we can see lots of structure, some hairs on the outside, and so on. Okay. This is stentor, which is another common eukaryotic single-celled organism. The cell is this funnel-shaped cell. It has the little cilia or hairs that beat at the top there, and it actually creates a current. And this is a predator. As bacteria and other things swim by, they get caught in that current, and they get sucked down into the funnel, and that's how they make their living. Okay. Giardia is a common parasite to humans. If we have problems with drinking water, it's usually because of Giardia. They cause lots of intestinal problems. Okay. Trypanosoma is another example. Here in this slide, all the red cells are human blood cells, kind of donut-shaped. The trypanosoma have been colored in gray, and they're like little microscopic leeches. They actually attach to the blood cells, and they would suck out the cytoplasm and the energy out of the cell. Certainly a trypanosoma infection is associated with anemia, lack of blood cells. Okay. And many eukaryotic cells are also photosynthetic, and so these volvoxes, each little sphere is a volvox, a colony of several hundred eukaryotic cells all bunched together. And when they reproduce, cells move to the inside and grow into new little spheres, and so we can see some of these volvox have lots of little spheres inside. Eventually the volvox will burst, releasing those spheres, and that's how they reproduce. So a lot of diversity within the eukaryotes as well. Now, it's one thing to say prokaryotic cells are small and eukaryotic are large, but what exactly does large and small mean? So if we compare these in the same slide, and this is just a shot from a light microscope, and the bacteria cells are all these little purple pill-shaped cells. And so they've been stained to be purple. They normally wouldn't be purple. But we can see in this slide there's probably 50 or 100 cells scattered all over. Our eukaryotic cell, though, is a large cell in the background here. And this is an animal cell. And we can see that this animal cell is 100 or more times bigger than prokaryotic cells. In fact, the nucleus of the animal cell, the little dark pink circle right in the center of that cell, is actually bigger than an entire prokaryotic cell. And so when we talk about small and large, these are very significant differences. But I also want to point out, just because our cells are larger or more complex, that doesn't make our cells better than prokaryotic cells. Being small and simple allows these bacteria and archaea to reproduce very quickly, to be very successful. To wrap up our cell introduction then, we compared our tools for looking at cells, our light versus our electron microscopes, some advantages and disadvantages of each. We've talked about the limitation of cell size and that idea of surface area to volume ratio, and that as cell size increases, that surface to area volume ratio decreases, and that's the main factor keeping cells microscopic. And then we talked about our prokaryotic cell characteristics, looked at a little diversity and some examples there, and then we finished off with our eukaryotic cells, their characteristics, and some examples of diversity there as well.